Okay, hello everyone. Thanks for coming to this talk, and thank you to the organizers for selecting our work for a presentation. And what I'm going to tell you today is some computational methods to try to understand evolutionary history of regulatory modules. So as you might know, a central question in biology is to be able to understand what controls phenotypic diversity of organisms. And it is becoming increasingly clear that changes in gene expression is an important player in controlling the morphological complexity of organisms. And with advances in genomics, we are now able to actually measure many different types of functional measurements, such as mRNA, protein, chromatin state, and comparative functional genomics is an emerging area that aims to measure these functional data across multiple species and systematically compare them to identify sets of genes, proteins that are modified in a coordinated way and try to correlate that to phenotypic traits. The challenge, so what we would like to be able to do is to be able to compare these genome-wide data across multiple species. Now, this is challenging because, as you heard from the first talk, mapping between species is actually not trivial because you might have gene duplications and losses. So now let's think about how we might do this in the context of, say, mRNA levels. A useful abstraction to think about when thinking about mRNA data is to consider transcriptional modules. So what are modules? Modules are defined as sets of genes that are co-expressed. Such modules can represent, for example, a pathway or a process that needs to be coordinately regulated. All the genes need to be coordinately regulated. What we want to be able to do is identify these modules in individual species, and we want to be able to ask questions like, what are the different patterns that these different modules exhibit across the different species? What do the genes that actually exhibit, what do they look like? Are they the same genes? Are they different genes? And more importantly, we want to be able to reconstruct how these different modules evolved. So you might realize that this is a clustering problem. It's actually a multi-clustering problem, one clustering problem per species. And then when we started off, there were some approaches that we could have used. So one idea is to actually make use of orthology information to try to concatenate the data matrices across the different species to make a horizontally long matrix and then cluster that to give you an initial clustering and then refine that based on species-specific clustering. So that's one idea. The other idea is if you actually have matched samples across different organisms, you can vertically concatenate them and make a vertically long matrix and cluster that. And this was soft k-means clustering, where another heuristic was put to be able to enable genes that share, which are orthologous, are actually in the same module. So put that as a heuristic. The challenge that we faced when thinking about these approaches was that they didn't explicitly take into account ancestry. And so if we wanted to reconstruct the module assignments, we had to do something as a post-processing step. And the second important thing that was missing was the ability to be able to handle gene duplication, because we were looking at many species, and we had to be able to work with a situation where there was not a one-to-one mapping, and so we had to work with these complex mapping trees. So what we did was we developed an approach, and so the rest of the talk is organized into 3 parts, where I'll give you a little bit of an overview of the Arboretum approach, which tries to tackle some of these issues. And then I'll talk about some work we did on yeast Ascomycetes. And then third, I'll talk about some very new work, which is actually unpublished, and I'm excited to talk about that, and I'm hoping to get some feedback. So what is Arboretum? So the key idea of this algorithm is to be able to use trees at the gene level, at the species level, to try to handle both gene duplication and also to be able to infer the ancestral module assignments. And it is based on a probabilistic process of how the data is generated. So what data? Gene expression data. So just to give you an idea of what the different components of the model is, there are 2 parts to it. The first part is associated with the module evolution process. And so this is an example phylogenetic tree with 3 species and 2 ancestral species. And for basically, as an input to the algorithm, we take the number of modules. So the module evolution is actually captured by what we call transition matrices. These are conditional probability distribution matrices, which essentially capture the probability with which a gene in a species maintains its module assignment or switches its module assignment from its ancestral species. Okay, and so we have this for every branch in the tree, and then at the root node we have an initial probability distribution of how likely a particular module was. So that's the module evolution part of the model. And then at the leaf nodes where we have gene expression data, we model that using a mixture of Gaussian models. So each component in the mixture is actually representing a module. So for every module, we have the mean and the variance that we would estimate from the data. So how does the process work? So generative models, as you might know, are based on, it gives you a recipe of how the data is generated. And so for every gene, we start with the most ancestral node. We draw a sample, which is the module assignment, and we propagate it down the tree. And then at the leaf nodes, what we have is a module assignment that points to one of the Gaussians from which we draw the expression profile. And so that's how we generate the data. And the way we learn the parameters, which are the means, variances, and the transition matrices, is through expectation maximization, which is a well-known algorithm when you have hidden variables, which are our module assignments. Okay, so that's the general framework, the algorithm, and when it comes to applying to data, there are some other steps. So one other thing that is novel about Arboretum is the ability to model gene duplications. So, again, the first talk did a very good job of explaining all of the challenges that arise when gene duplication happens. So let's assume that you have a gene duplication between species A and B. The question is, what do you do in this kind of a situation? So assume that CAI is the module assignment for the ith gene family. So we need to think about how the module assignment will be evolved for a gene tree which has a duplication. So in the case, on the branch where there's no duplication, we just evolve the module assignment in the same way as we did before. However, when we have a gene duplication, until the point of the duplication, we do the same thing. However, when we reach the point of duplication, we just draw 2 samples of the module assignment, and that fits naturally inside the model. So this is how we actually handle gene duplications. And so we make use of the topology of the gene tree to be able to just incorporate that inside the model. So the way Arboretum works is we give it as input gene trees and species trees and gene expression data across multiple species that you're interested in. And then as output, it will give you the module assignments, and then it will also give you the parameters that it learned. And then these can be then examined for different types of Go enrichment or motif enrichment. And you can also use these inferred module assignments to actually study known pathways. And you can actually query the inferred module assignments to be able to see how these evolved across in a particular phylogeny. And then we have some metrics that we use to try to quantify how much a particular gene or a module changed. So I'll tell a little bit about that. So what we did, so this particular phylogeny was the original motivating example for this approach. And so I'll just give you a quick tour of this phylogeny. This is 15 yeast species, and they span about 300 million years of evolution. And it's a very interesting phylogeny. So some of these species are called respirofermentative. What that means is that in the presence of glucose, they like to ferment. And it includes Cerevisia, which you might know is a very well-known model organism. And then another set of species are respiratory, which includes one of the human pathogens, Candida albicans. These are respiratory. And these 2 species are separated by the whole genome duplication event. So there is this major event that happened in this particular phylogeny. And then at the very end, we have the respirofermentative life cycle coming again. So basically, there is this complex phylogenetic structure out here. And we wanted to use this to study regulatory evolution. We want to use this as a model to study regulatory evolution in eukaryotes. So what we did was, first of all, to decide what is it that we need to measure the expression under. So, so this is an important question. So what, because that can influence a lot of your results. So the criteria for us was to be able to measure enough, so there would be enough variation across that we could actually study. So glucose metabolism was one condition that was good to look at because of the inherent dependency of the species on glucose. And the other conditions that we looked at were a bunch of different stress conditions, and this was for a subset of the species. So in general, we have like all of these different stress-related data sets for all of these different species. And what we did was we first applied the glucose depletion data to Arboretum, and what we inferred were modules. So what I'm showing you here is the result of applying Arboretum to these 15 yeast species. And what you see here are 5 major patterns of expression, 5 modules. And these modules are actually numbered from 1 to 5, and they exhibit a very conserved pattern of expression. And these are color-coded based on microarray terminology. So green means repression, red means induction. And if you look at the genes that are within each of these modules, they're associated with the repressed module associated with growth-related processes, and the induced module is associated with respiration and stress-related processes. So, so basically at the pattern level, we see a lot of conservation. However, this does not necessarily mean the genes within each of these modules are the same. So we need to explicitly measure that. And what we do is we take, for every pair of species, we compare every pair of modules, and we ask, what is the extent of gene orthology overlap between these pairs of species? And we compute this matrix, and we color-code it based on the extent of orthology overlap. So along the diagonal, we are basically saying those modules that have the same pattern, how similar they are in terms of the gene content. And then the off-diagonal elements are basically modules that might have actually changed their genes. So we computed this matrix for this phylogeny, and what you're seeing here is the comparison of Cerevisiae with all the other species. And what you see is a strong diagonal element. So in general, there's a lot of conservation, but you start seeing these blue elements, which suggest the modules keep diverging, and this increases with phylogenetic distance. So we did this for the entire 15 species, and you see as you go down further away from the species that there is this increased divergence. So the genes generally exhibit similar patterns of conservation. The species exhibit similar patterns of conservation, but there is divergence which is correlated with phylogenetic distance. So this was all happening at the extant node. Now what happens as far as the ancestry is concerned? So as I mentioned before, Arboretum infers the ancestral module assignments, and it is done during the process of learning the modules. And what I show you here is a particular tree for a subset of these species for a particular gene called the NIP7 gene, and I've basically color-coded the inferred module assignments. And what we do is we just represent this as a string of numbers. This is just the result of doing a post-order traversal on the tree. And we do this for sets of genes just to see how sets of genes actually change their pattern across evolutionary time. So in particular, this is an example of the most repressed module in the last common ancestor of all of these species. And what you see are genes along the rows, and columns represent species. And what you see is this general pattern of green, which suggests there's generally a lot of conservation. And you see a similar picture for all of the different modules. So this is the first, second, third, fourth, and fifth, where generally the matrix is of the same color. However, there are places where these genes have actually changed their module assignment. This is a better pointer. So for example, here you see that these genes, they have the same sequence, but they have actually changed their modules. And these are very interesting genes because they could tell us something about these species and how they've evolved. So one particular example was that of these genes that are associated with mitochondrial, which are enriched for mitochondrial ribosomal proteins. So these genes exhibit this interesting pattern where the respiratory species are all red, whereas the respiratory species are all green, that is, they're repressed. And using Arboretum, what we inferred was that the most ancient ancestor of all of these species was respiratory. So, and this was consistent with what would be expected from the literature. And so this was a nice validation of one of the predictions of the ancestral module reconstructions from Arboretum. We also used Arboretum to try to understand how different processes evolve. And so, for example, we applied Arboretum to a subset of the species for which we measured heat shock. And what we found was that the patterns were very similar. However, the modules themselves were actually not very similar. And in fact, we found that heat shock was less conserved. So what you see here is each circle is color-coded based on the module it actually represents. So green means very repression, strong repression, and red means induction. And basically, the x-axis represents the extent of gene content overlap between modules in heat shock, and the y-axis represents the extent of gene content overlap between modules in glucose depletion. And what you see is pretty much all of the circles are below the diagonal, which suggests that glucose depletion is more conserved. So hopefully, this gives you a flavor of some of the things that we did. And then we also looked at gene duplication. And as you might know, there are several factors that influence how genes actually, species acquire new traits, and gene duplication is one of them. So one question we asked was, can we say something about, we see all of this divergence, is there any, can we say something about gene duplication? And what we did was we defined a measure called module reassignment index, which is simply the number of times a gene changes its module assignment. And we compute this for each gene tree. And what we found was genes that have a gene duplication, or gene families that have a duplication, tend to change their module assignment much more. So they're likely to diverge their module assignments much more than those that don't. So just to conclude from the yeast study, what we found what we did was we developed an approach to try to study modules across multiple species. And what we found was that there was generally a lot of conservation. However, the extent of conservation decreased as a function of phylogenetic distance. And we found genes that would change in a phylogenetically coherent manner, or in a manner that was associated with the lifestyle of these organisms. And when we found that gene duplication was an important factor that would control, that would play an important role in the divergence of these modules. So now I'm going to shift gears and tell you very briefly about what I've been doing transitioning from yeast to mammalian systems. This is basically published data from Rowan et al., which is one of the very comprehensive studies actually looking at mammalian gene expression in 9 mammals and chicken. And they looked at these 6 tissues, and they measured gene expression using RNA-seq. So this serves as a good data to see how well Arboretum performs on mammalian data sets. And just to give you a sense of what the data looks like, we used ensemble comparer to get gene trees, and you can see that as you increase the number of gene duplications, you actually get more and more and more gene families that you need to look at. And this is work that I'm doing with my postdoc, Sarah Knack. And all the results that I'll tell you is basically when we look at our orthogroups, which have only one or no duplication. Okay? So as we did in the yeast case, we applied Arboretum, and here we had actually 7 modules, and these modules are ordered based on the expression level in the brain tissue in human. And so basically it goes from green to red. As in general, we see that there are certainly patterns of conservation that are actually present in that you see out here. However, it's not as clean as we saw in the yeast data. So certainly there is place for improvement. Now, what we're doing is trying to annotate these modules based on the pattern of expression. So for example, the first module, what I've done is I'm showing you their expression patterns, and from human to chicken. And what you see is there is this conserved induction of these tissues, of genes in these tissues, and these are actually associated with liver and kidney. Similarly, we see that there is another module, which was on the other extreme, where we see very conserved induction of brain and cerebellum associated tissues. And then finally, we are actually still mining this data, but one of the things that you can ask is query this data for known pathways and gene sets. So one interesting example is to look at, for example, transcriptional regulators. And these are just transcription factors. And what I'm showing you is an example of a particular transcription factor, which it's actually not a transcription factor, but it's MECP2. It binds DNA binding, DNA methylation, DNA which is methylated. And it is associated with several neurological diseases. It is highly induced in human and is not as much in the other species. But anyway, I just want to conclude with what I told you today, a general framework to try to look at modules, to compare expression data using the abstraction of modules across species. And of course, the next steps are to try to use these kinds of approaches and extensions to think about regulatory networks and how they evolve. And with that, I want to conclude. This was work that I started with Aviv and Manolis as a postdoc. And I want to thank Don and Mike. They are yeast and mammalian collaborators. And Sarah Knack, my postdoc. And I just want to take this time to advertise my student's poster, who's actually going to be talking about something completely different. So please come check it out. With that, thank you, and I'll be happy to take questions.
So the ancestral yeast genome underwent a whole genome duplication. I think it was, 200 million years ago or something? And it's previously been observed that when that happened, the sequence constraints on the retained duplicates went way down. So when you look at my actual question, so you said that the phylogenetic distance between the modules was very predictive of the level of conservation in the module. Was that independent of the sequence conservation level of the genes in the module?
The module conservation is computed based on the genes that are inside those modules. So
right. So if you take a module and you go human to worm, right, then the module is more scrambled, and also there's a lot of loss of sequence identity. Right. Whereas if you go human to chimp, there is less scrambling of the module and less loss of sequence identity.
Right.
Is the increased scrambling as you go from human to worm on a per module basis explained by the differences in the sequence identity between the genes in the modules or not?
That's a good question. We haven't systematically actually looked at that, so all I can tell you is whether there was conservation or not. But I don't know. I mean, maybe we can talk about that a little bit, but you know, I can't break it apart. I can just tell you that there was conservation or not.
It's difficult to compare that with what's already known in yeast if you don't have
that. So I've heard a couple of theories for how you would evolve a co-expression module, and one is effectively that the transcription factor binding sites evolve, and the other is that the transcription factor evolves. And I think those would lead to pretty different patterns of evolution. I was wondering if your results lead to any insight into which is more common, or do they both occur?
Yeah, so cis and trans. I didn't get a chance to actually talk about any of those results. We see, actually, what I did was I looked at the expression levels of the transcription factors to see the extent to which it actually changes. At the level of gene expression, we don't see a lot of changes. So and you know, with the cis regulatory elements, we did some motif enrichments to try to see, you know, do we see a conserved it can be actually explain much of this divergence based on what motifs are gained or lost. And we didn't do this at the module level, but actually in Aviv's group, they actually did this, and they found that there's a lot of movement happening at the cis regulatory state, but there are actually constraints that are maintained more at the module level. I think this is a roundabout way of trying to get to your question, but I think there's a lot of changes happening. There's more changes happening at the cis level than the than at the transcription factor level. However, what we need to be able to do, I think, which is another direction that I want to take, is to actually have better ways to find motifs across species, because that's, and maybe it's not actually individual motifs, but maybe cis-regulatory modules which actually somehow take into account interaction between different transcription factors, and try to see if that can actually somehow explain the divergence in the different modules.
Hi, Taylor Rayborn, Indiana. I really enjoyed your talk. I just had a quick question about the last bit of results that you presented about sort of the pan-vertebrate comparison that's ongoing. What data did you use to do that? Because in my own experience, and I'm doing something related but not precisely overlapping with this, I'm looking particularly at promoter architecture, and I find that there's asymmetry about the amount of data that's available. There's a ton of data for mouse and human, and then it's more sparse across other taxa. I wanted to know if that was your experience, or if that could have explained some of the result that you saw when you found that you couldn't actually, you didn't see as much consistency or confirmation across. So.
Right. So there are two types of data that we're using right now. So gene expression and sequence. Yeah. So the sequence data is pretty it's been there for a while. However, one can wonder about the quality of the gene trees. So that's one of the problems that we have to kind of worry about. So as far as gene expression data that I used is actually published data from Browand et al. And you're right, human and mouse is actually much more studied. But when you're actually learning those modules, you probably want to only use those data that are coming from the same experimental condition, just to make sure that when you're seeing differences, it's not because you took data from different places. So that's like the expression data, which is uniform for all of these. And they did a lot of quality controls to make sure the data were comparable and all that. But the problem happens when you're trying to interpret these modules, and there you would want to interpret it in the context of human or mouse. And
yeah, that was my question. So you use a single data set that was from a single study, or the data sets from a single study?
Yes, from Browand et al. Yes.
And Rowan et al.
David Browand.
Okay.
It's a Nature paper from 2011.
Perfect. Thank you.